Today we're going to be talking about how neurons communicate, which relates to action potential. So as you can see, there's a picture of a neuron. I just put it up there to recap. So you have dendrites, cell body, uh, axon, axon hillock, and the axon terminals. So when we talk about cell communication, neuron communication, it's important to understand the concept of action potential. The action potential is driven through the membrane potential, the electric charge, and the axon. So the membrane potential results from the balance between two opposing forces, the diffusion, where molecules distribute themselves evenly throughout the medium in which they reside, and the electrostatic pressure is when particles with the same electrical charge repel one another, one particles with the opposite charge attract one another. So the environment inside the axon and in the fluid surrounding it contain different ions. And predominantly, mostly outside the ion, you have chloride ion, Cl-, and sodium ions, Na+. And mostly inside the axon is potassium, K+. And only found in the inside the axon, organic ions that are also proteins. So in this chart, you can see the resting potential, threshold, depolarization, repolarization, and the absolute refractory period and the relative refractory period. So when the cell membrane is stimulated, the sodium ions that are outside the axon enters the axon through sodium pumps. And this entry of sodium ions disturbs the resting potential and causes the inside of the cell to become more positive relative to the outside of it. So this depolarization at the membrane leads to action potential. So it's a very brief reversal of potential, and the action potential is responsible for electrical signaling and information transfer in the nervous system. So it's very important. So the threshold is the critical level to which the membrane potential must be depolarized in order to initiate an action potential. So minus 55 megawatts represents the threshold potential. And beyond this, we get a full action potential. So the membrane potential rises to plus 35 megawatts. So that's the peak of the action potential. This is the small fact that you should know. The area, the region of the neuron that has the highest density of voltage-gated sodium channels are at the axon hillock. So that's where the action potentials initiate. They start from the axon hillock and travels all the way to the axon terminals. And the electrical signals in the dendrites and the cell body of the neuron is called graded potential. And one other important thing that you should know is that action potentials are of the same intensity. Every action potential are the same intensity. Stronger sensation result in more impulses, not strong impulses, they are all same. And more impulses from the same neuron, and the more impulses from the same neuron cause an intense amount of action potential. So the refractory periods, the absolute refractory period is when membrane cannot produce another action potential because the voltage-gated sodium channels are inactivated. This is because they're still open or they're not responsive. The relative refractory period is when the voltage-gated potassium channels are still open, and it makes it harder to depolarize to the threshold. So The axons with the myelinated axons are much faster in transporting the signal than the unmyelinated axons. So the proper communication part, the functioning of the brain involves the flow of information through networks of neurons, so the wire-like branches. And the neurons receive information from hundreds and thousands of other neurons through little junction points, and these are called synapses. And in these synapses, you can release neurotransmitters, those chemical signals that releases the information. So to be a neurotransmitter, it must be produced and stored in the neuron and be released when the neuron is stimulated, so that when it's depolarized, and it has to bind to postsynaptic receptors and have an actual biological effect, and be inactivated by degradation, uptake, and metabolism by an adjacent cell or reuptake by the presynaptic neuron. And it also should mimic endogenous activity by exogenous application to the neurons. So some examples of these neurotransmitters can be serotonin, dopamine, and glutamate. And same neurotransmitters can have different or opposite effects. So the process of which the neurotransmitter molecules detach from the postsynaptic neuron can be where it's reabsorbed by a presynaptic neuron, so they can be recycled and used again. This process is called reuptake. And the neurotransmitter binding to receptor in the postsynaptic neuron could lead to either depolarization, so the excitatory synapse, or hyperpolarization, which is the inhibitory synapse. And a lot of postsynaptic potentials, synapses, are summed up within one neuron. So thank you so much for listening. I hope to see you in the next video where I'm going to be focusing on the hypothalamus. So see you next time. Bye.